So here we've got the lesson 10 group problems. Question 1, suppose a eukaryotic messenger RNA has the following nucleotide sequence. Got that right there. Assume that RNA polymerase II produces this molecule from right to left. So we've got the 5' end on the left and the 3' end on the right. It says, what is the sequence of the template DNA strand? So the template DNA strand is going to be the pairs of that RNA molecule in DNA, which right here we have ACG, AA, CGG, TTT, AGT, GGG, CTA, TCT. And then C, what is the sequence of the coding DNA strand? Include polarity. So that one will match the RNA with the 5' on the left and the 3' on the right. And it's exactly the same as the RNA molecule, except the U's are replaced with T's. Keep that in mind. So D, draw an image that shows the 3 nucleic acid strands above during the elongation stage of transcription. And be sure to label the mRNA, the template DNA strand and the coding DNA strand, and include their polarities. On which side, to the left or the right of the template strand, is the transcription start site and the top box located. So it's going to be on the right side in this drawing here, on the left side. And I use different ones so it fit better and you could see it better. I use different nucleotides. So you have the coding strand on the top and the template strand on the bottom, with the RNA here in the middle matching the coding strand opposite that has been opened. So question 2, fill in the blanks. A, sigma factor binds directly to the TATA box, while TF2D binds directly to the 10 and 35 sequences. B, guanylyl transferase is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide. And the group 1 introns use the nucleoside guanosine as a cofactor during catalysis, while the G2 introns use adenine as a cofactor during catalysis. And D, CPSF is an endonuclease that assists in terminating transcription in eukaryotes. E, in lesson 9 we learned about general transcription factors. TF2B is a general transcription factor which acts as a bridge subunit to link TF2D to TF2F RNA polymerase II. And TF2H is a general transcription factor which acts to phosphorylate and activate RNA polymerase II. Finally, F, the spliceosome is composed of subunits called SNRPs. These spliceosome subunits are composed of 2 components, proteins and small nuclear RNAs.
This is James. Leo, I'm doing questions 3 and 4 of lesson 10 group problems. So question 3A asks us to indicate 2 proteins in either prokaryotes or eukaryotes that act as helicase to separate 2 DNA strands during transcription, and that will be TFH2H subunit and RNA polymerase. B, indicate 1 protein in prokaryotes that acts as helicase to separate RNA from DNA during transcription, that will be the Rho protein. C, indicate 3 molecules in eukaryotes that function during RNA modification as ribozymes. That would be small nuclear RNAs, pre-mRNAs, and snRNPs. D, indicate 1 protein in prokaryote that acts as transcription factor. That will be sigma factor. And finally, E, indicate 1 enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides but does not use template DNA strand for RNA synthesis. That will be reverse transferase. Question 4 asks us to draw images of nucleic acid structures after hybridization experiments that was described. A asks us to draw the pre-mRNA for YFG gene that is hybridized to the genomic DNA copy of YFG. And so here is my representation of the genomic DNA strand on top and also the RNA with the 4 introns. B asks us to draw the mature mRNA for YFG that is hybridized to the genomic DNA copy of YFG. So again, the top is the DNA strand, the bottom is the mature RNA strand. So as you can see, these are the R loops for the 4 introns. Once we take it out and combine them, this is what we get for the mature RNA. For C, the mature mRNA for YFG is hybridized to the genomic DNA copy of YFG. In this case, the 5' most intron in the pre-mRNA contains a defective donor site and branch site adenine. Assume that the introns are removed using the spliceosome method. So the 5' most is defective. In this case, that will be intron 1. So it's still there, but it doesn't form an R loop like intron 2, 3, and 4. The question 4, the mature mRNA for YFG is hybridized to the genomic DNA copy of YFG, in this case the 2nd and 4th introns. So it's a similar question, but 2nd and 4th. So in that case, they will still be there, but they won't form R loops. And once the DNA is spliced, this is what we'll get for the mature RNA. So that's questions 3 and 4.
Hey, so it's Dylan. I had problem number 5, and it says below is a list of proteins and nucleic acid sequences involved in transcription and RNA modification, and then it gives you the list. And so what I did was I just put letters down for what applied. So A was which of the above is found in the bacterium E. coli. So going back through the notes on E. coli, I know it's the RUT, the group 1 intron, and the NUSA. And then B was which of the above is found in the yeast Saccharomyces cerevisiae. It was pretty much all of them except the -35, the RUT, and the NUSA. For part C, which of the above is involved in transcription initiation? So that's going to be the -25, the enhancer, -35 as well, GC box, TFIID, and the silencer. Part D, which of the above is involved in transcription termination? That's only the RUT and the NUSA. And then which of the above is a DNA sequence? And that's going to be the -25, the enhancer, -35, the GC box, and the silencer. The other ones are just proteins.